Hello, my name is Ricardo Velázquez-Aponte, and along Dr. Celine Cassé at the University of Puerto Rico, Mayagüez, I am working on the antiproliferative properties of ethanolic and aqueous graviola leaf extracts on tongue squamous cell carcinoma cell line SCC25. SCCs, or squamous cell carcinomas, are types of cancers that arise on the epithelial tissues of our bodies and account for 90% of all cancer cases in the head and neck cancer group, which comprises cancers that arise from the mucosal cavity all the way down to the pharynx and larynx. Common treatments for these types of cancer include harsh chemically invasive therapies, such as chemotherapy and radiation, which have been shown to present a very low specificity between healthy untransformed cells and cancer cells. So, as therapeutical alternatives, our project moves to natural products, specifically Annona muricata, commonly known as Graviola or Guanabana, which is an evergreen tree native to the subtropic region, which anti-proliferative properties as well as anti-inflammatory properties have been very well documented. The purpose of a study is to assess the anti-proliferative properties of Graviola extracts using an in vitro model of SCC25 cells. In order to do so, we performed a set of extractions, where the first one was an ethanolic extraction, where we used pure ethanol as our solvent, and we were able to obtain a crude graviola leaf extract in a liquid form. And our second extraction was an aqueous extraction, where we used purified water as our solvent, and we were able to obtain a solid powdered extract. We then performed some cell viability assays, which is what you will be able to see here in table F, where we present the results of a dose-response analysis in which the SCC25 cells were treated with increasing amounts of the ethanolic or aqueous extract for 24 hours. And here is also detailed the GI50 value, that is to say, the corresponding dose responsible for exerting a 50% growth inhibition in our cell population. And what is interesting is that if we compare the GI50 value of the ethanolic extract to the GI50 value of the aqueous extract, we can see that the ethanolic extract showed a 4 times higher antiproliferative activity on SCC25 cells. And to better corroborate this antiproliferative activity, we performed some dose-response assays, which are presented here in figure E, where we treated the cells for 24 hours with 100 micrograms per milliliter of each of the extracts and a positive control to visually assess the proliferative activity. You will be able to see here in box T2 labeled EE, that is to say the ethanolic extract at 100 micrograms per milliliter dose. When you compare box T2 and T3 labeled AE for the aqueous extract at each corresponding concentration, you can see that the ethanolic extract substantially affected the cell viability on our cells, and which was expected since we already know that the GI50 value was lower than the dose administered here in this example. And from also this image, we can see in row N, where we administered a 0.1% volume by volume concentration of each of our vehicle solvent, we can see that the cell viability was not affected by any of our solvents. This tells us that the antiproliferative activity we were seeing was in fact due to the phytocompounds extracted on each extract. Specifically on the ethanolic extract, where we see the more antiproliferative activity, which is why we hypothesize that the antiproliferative properties of the Graviola leaf extract are imparted by the phytocompounds extracted in the ethanolic extract. Keeping this in mind, our future plans involve the characterization and the isolation of these biocompounds. We also plan to test and compare the in vitro toxicity of the ethanolic extract on another SCC cell line and assess its activity on healthy oral cells, as it has been shown to be the case in other botanical extract and crude plant extract, that there is a lower toxicity towards healthy sane cells than it is to cancer cells. We like to thank the Puerto Rico INRAE program for their support and the Camtox facilities for the cell viability assays, as well as Professor Laura Lopez Moreno at the chemistry department of UPRM for her help with the extraction processes. We hope you have found this information helpful and thank you for your attention.